DNA is the genetic code of life, so it's very important for us to learn about the structure of DNA and how it functions. As a nucleic acid, DNA is made up of several nucleotides put together. Each nucleotide has a phosphate group, a deoxyribose sugar, and a nitrogenous base. DNA is shaped in a double helix or a twisted ladder. The sides of the ladder are made up of the sugar-phosphate backbone, while the steps of the ladder are comprised of the nitrogenous base pairs. The bases are matched in an anti-parallel way and are bonded by hydrogen bonds, which are very weak. The four bases of DNA are adenine, thymine, cytosine, and guanine. Adenine always pairs with thymine, and cytosine always pairs with guanine. Because of this, Chargaff's ratio states that the percentages of adenine and thymine always match, and the percentages of cytosine and guanine always match. A always matches with T, and G always matches with C. Since DNA is so important, it must replicate before a cell goes through mitosis, so that each cell can have identical genetic information. To replicate, DNA must go through 4 steps. The first step is to untwist the double helix, and secondly, it must break apart the hydrogen bonds holding those nucleotides together. And this is done using an enzyme called helicase. Next, we need to add new nucleotides to each old strand using an enzyme called DNA polymerase. Then, the nucleotides are glued together using DNA ligase. DNA replication is called semi-conservative because it results in 2 identical copies of DNA, each with 1 old strand and 1 new strand.